to think about that. But let's first go to the action potential. So we want to take into account, I'm just going to read this table first, and then we'll go through this in the same context we just did, asking about at any one point in time. The x-axis is time in milliseconds, units of milliseconds, and the y-axis is membrane potential in units of millivolts. And we see it goes from minus 90, which is what? That's right, the equilibrium potential for potassium, so that's why we see it there. And plus 30, which is closer to the plus 60 equilibrium potential for sodium, the action potential does not reach that. So we'll read through this and then interpret each portion of the action potential in that context. So there we'll see the resting membrane potential, which for neurons classically and globally is considered to be on average minus 70, but it will differ from neuron to neuron. It definitely differs from cell type to cell type. Muscle, skeletal, and cardiac might differ, and then across species too. There's going to be a depolarizing stimulus, some type of initial event, a graded potential is what we typically would think. Then there's going to be a membrane depolarization to threshold at the trigger zones, such or in a localized portion of the axon. I like to think of it physiologically starting at the trigger zone. That's my perspective. The voltage-gated sodium channels open quickly, and sodium enters the cell. The voltage-gated potassium channels begin to open slowly. So here, both of these channels, the voltage-gated sodium and the voltage-gated potassium channels, are sensitive to voltage, and they are voltage dependent for opening, but their rate of opening differs. So their stimulus to open is the same, but their rate of opening is different. It's rapid for sodium and slow for potassium, so their peak effects on the membrane potential are going to be offset in time. They're going to begin at the same time, but their peak effect over their dynamic range, their temporal dynamic range, is going to be offset in time. And then we'll see that the stimulus for closing differs too. It's time dependent for sodium and voltage dependent for potassium. So you should write those down, and I'll say them one more time so you can write them down now. So we have opening, the rate of opening, and closure. So the sodium and potassium are both voltage dependent for opening. The sodium channels, voltage-gated sodium channels, rapidly open, and the potassium channels slowly open. The voltage-gated sodium channels, the sodium channels are time dependent for closing, and the potassium channels are voltage dependent for closing. So there's a rapid sodium entry into the cell that depolarizes the membrane. The membrane potential deflects toward the equilibrium potential for sodium that we said was plus 60. So it's going to pass 0 and approach a positive value, and it gets up, as you see, obviously, to about plus 30. And then that's the time period that the sodium channels have to be open. Now remember, they don't open, it's a population event. Not every single one of them opens at exactly simultaneously the same time. They're offset by a bit. So this is a population-created event. So it is gradual over time. So gradually, if we look at the curve here between 2 and 3, gradually that rate changes, and then pretty much they're all open at the same, maximally open, but some are already partway through their time period that they're going to close. So somewhere along here, the first ones to open start to close, and then the later ones that opened begin to close. They have a certain period of time that they're going to stay open once they're opened. But by number 5 here, they all will have closed. So they all don't open at the same time. Just picture a group of people standing in one area. Remember, this is just the change over time in one single spot. So you're looking at a group of people in one spot, in one circle, and they're all going to open their mouths and scream for a set period of time. But they all don't start at exactly the same time, and they all don't end at exactly the same time. But there is a time where they're all going to be screaming, and then some will start to stop, and then finally they all will stop and there will be silence again. So that's what's happening here, is a dynamic population event. And I don't like these reduced images that don't give you that feel for that population event. You don't really understand physiology if you don't appreciate that population event, because this is where probability comes into play. What is the likelihood that you're going to observe this event at any one point in time? I hope that helps you to understand that. So the sodium channels close, and then the slower potassium channels are going to begin to open. Then we're going to see a change in the deflection of the membrane potential, but in what direction? The Nernst equation, therefore, would predict that we would see a hyperpolarization, a hyperpolarizing event. Actually, there's a repolarizing, but it's still in the hyperpolarizing direction as an overshoot. So that's the number 7 over here. Until what? Until some membrane potential is reached that will begin to close the voltage-dependent closing of the potassium channels. So along this curve, picture the voltage-dependent sodium channels opening and closing, rapid open, time-dependent closing, and then the potassium channels slow opening and voltage-dependent closing. And I think we do have that next one to take into account. And then 9, the cell returns to the resting membrane potential. Okay, so what is the state of the sodium channels throughout this from point 2 to point 9 that we'd say is really established right about here? Right, so of that whole population that we just, the people in the circle, after they shut their mouth, then they've got to kind of, ruffle their shoulders and rub their cheeks or whatever to get reset to where they can open their mouth again, right? But there's a population of them. So over this period of time to where they stop shouting, to where they all are available, some of them are going to be available to open their mouth and shout again, right? So we want to think about that, where there's a time point where none of them can do it, to where some of them can do it, to where all of them can do it again. And this helps us understand this refractory period in the two portions. There's an absolute and a relative refractory period. So it's not shown on this one, but we'll talk about it ahead of here. But I just want you to appreciate that. So now let's go along. Resting membrane potential. There's some event, some stimulus that causes an increase in sodium influx, so a current into the cell, and that is sodium. So we get a deflection in the membrane potential towards the equilibrium potential of sodium. Is it sufficient to reach threshold at the spike zone, or at this level, in this area of the axon, where the density of voltage-gated sodium channels requires this level of membrane potential difference before they will open sufficiently to cause a regenerative effect where they'll open more guys near them and they all get shouting, right? So they all start to shouting. They're elbowing each other saying, you shout now, right? and they all start shouting together, and the first ones to begin shouting, first ones to open their mouth to start shouting, are the first ones to stop shouting and begin shutting their mouth and shrugging their shoulders and rubbing their cheeks, until ultimately somewhere along here, those first ones to open here are ready to be opened again, and the later ones to open will have been closed by this time, but are going to require this time out to here to be ready to open and shout again. So that population thing is really important. So this is sodium, and then once all of the shouters have stopped and closed their mouth, the voltage gated sodium channels are inactivated, as it's called. They've shut their mouth. No more sodium can come in. So then you are no longer going to have the membrane potential deflect in the other direction. But at this time point, you've got enough potassium channels open where enough potassium efflux is going to occur, and the potassium moving out of the cell will repolarize, and once the lower than resting membrane potential or hyperpolarized membrane level is achieved to trigger the voltage-dependent closing of the population of potassium channels, you'll get a gradual closing. Those that first, well, actually, it isn't based upon who first opened and who who second opened. It's really just a population event that as they begin to experience the local membrane potential, they're going to respond as a population and gradually reach a point where they are all closed and there's no more potassium efflux occurring. But remember this gradual recovery here also, as you probably had been correcting me throughout and is not shown here, the sodium-potassium ATPase pump is going to be more effectively moving sodium and potassium through this time period, because the sodium-potassium, more of it is there right near the membrane to move. So I tried to be very careful to say more of the sodium and potassium right near the membrane is available to be moved, because we know that there's no change in concentration of potassium inside or outside, or sodium inside or outside, except just adjacent to the outside or inside of the membrane, correct? So wording is very important in this type of study. Now let's think about the superimposability of the ion permeability. So one axis, the other axis over here, should be labeled in membrane potential in millivolts for the dashed line, correct? So we know that this is the plus 30 and this is the minus 70. So the after hyperpolarization, as it's called, this is called the after hyperpolarization. So it's after the action potential event, and it is a hyperpolarizing event, so it's the after hyperpolarization. So in more advanced studies of neurophysiology, you'll find that the after hyperpolarization, the potassium channels can be affected so that this can either be exaggerated and extended, or sometimes even shortened, so that the relative refractory period can be affected. So realizing that there's a variety of potassium channels is really important. When we talk about the cardiac action potential, that will become very important there as well. So it's not just one type of potassium channel that's contributing to this smooth biological signal, okay? But it is potassium efflux, no matter which type of potassium channel is doing it. When it's open, potassium's going out. Okay, so this shows us the after hyperpolarization that we just talked about, and the resting, rising, and falling. So the depolarizing, the peak, and the repolarizing, the rising and the falling. then the after hyperpolarization, and then the return to the resting membrane potential are shown in this temporal scale from the left to the right. So what we want to focus on in this one, again, is the ion permeability. So the orange, gold-colored curve is the sodium. So this is permeability. So if we looked at conductance, it would be basically the same, right? So this is the permeability. So that's the likelihood that sodium would move if it was present, right? The conductance would be a measurement of the actual movement of the flow of the sodium. If you measured current, then you would see an inward current here that would be made up from sodium, and an outward current here that would be caused by the efflux of the potassium. So I'm hoping that you can put together these ideas of permeability being one factor. We talk about it here because of the Goldman equation. It's easier for me to call it that. But whenever you have permeability, if the ions are present, and biologically they are, but if you're in another in vitro system that you've created, it could be something totally different than normal levels biologically. So if you have permeability and the ions are present, you're going to have a conductance. If you have a conductance, the flow, then you're going to have a current that can be measured. And the current is in amplitude, amps. So typically milliamps or microamps is the current that would be measured at a membrane. And so is it an inward current or an outward current? So we see here these two waves, and this is why conductance or permeability are shown on this type of a curve along with the action potential, because everything goes in the same direction. So it kind of makes sense. As permeability to sodium increases, or conductance, then you get the Nernst equation predicting that we're going to go toward positive 60, right? If you think of the Goldman equation, the permeability is going to increase for the sodium parameter selectively, so it is going to increase the membrane voltage, the membrane potential, and when those sodium ion channels close, permeability is going to decrease, conductance is going to decrease, the current would also disappear, and then that will fall back to the resting level of permeability, because the voltage-gated channels are going to close and reset. And it's a population event, so they don't all do it. They don't all do everything at exactly the same time, so you're getting this smooth biological curve, right? If everything happened at exactly the same time, you would get a true square wave, although the top of the square would be very short, and it would just be straight up, straight over, straight down. But it isn't. It's a biological event dependent upon a population of proteins that are changing, with each having specific biophysical properties: voltage-dependent opening, temporal dependent closing, and you see that the population of them all having temporal differences of starting and stopping then produces this smooth biological curve. Same thing for the potassiums. And so because the potassium channels have the same voltage sensitivity for opening, they begin to open, but at the same time as the sodiums, but their permeability is slower, so the slope of the tangent to this line is less than the slope of the tangent to this curve, right? Remember any of your calculus related to of course, this is a linear portion, so if you just took the slope of this linear portion, it would be a high number. So the change in y over the change in x would be very high here, and it would be much lower here. So the slope, the rate, because this is time, it's a change in y over a change in x, and if x is time, then that's the rate. So the rate of change in the sodium permeability is very high, whereas the rate in the change in the potassium is lower. And then when you get the negative slopes for the decrease, you would say the same thing, correct? And so it is the biophysical property of the slow rate of opening of the potassium channels that offsets the timing of peak values of increased permeability, and that conveniently creates a potassium efflux to hyperpolar to decrease membrane potential toward the minus 90 equilibrium potential of potassium. And because they stay open until the voltage is appropriate to trigger their closure, you get the after hyperpolarization. So you have no sodium conductance or sodium current out here, but you still have some potassium. And if you have a potassium conductance with this permeability, it is an efflux, it's an outward current. If you have the outward current of potassium, you get the membrane potential approaching the equilibrium potential minus 90 millivolts of potassium, which it does. And so the potassium current itself is going to produce an electrical membrane property that is sufficient to close its own voltage-gated ion channel. So it's a negative feedback system. So the voltage-gated potassium channels have a negative feedback system. They regulate themselves. As long as the biology is appropriate, it will function that way. The sodium dependent channels, the voltage-gated or voltage-dependent sodium channels are timed events. You can't have those. You can't take any chance of letting them regulate themselves. Is another way of thinking about it. Okay, so let's look at that voltage-gated sodium channel specifically in this illustration. So here's the sodium channels having two gates, an activation and an inactivation gate. So evolutionarily, it would seem, or we could kind of say, that you just can't take the chance to let sodium regulate itself like you can the potassium. In other words, the organisms that expressed this type of protein survived and reproduced, and this is the system that evolved for the voltage-gated sodium channels. The protein, it doesn't look like this, it isn't really a little ball, but there's a portion of the cytoplasmic segment of some of the proteins involved in this sodium channel that is on a hinge-like structure. And what happens is that there is within the transmembrane spanning segments of the proteins for the voltage-gated sodium channel, there are amino acids that are sensitive to the potential difference across the membrane. And note the proximity, the close proximity of the positive charges outside, the sodiums, and the negative charges inside, the anionic proteins, to the membrane. So it's just this immediate differential, charge differential across the membrane. I don't care what it is way out here or way out here, other than that concentration difference being important for the chemical gradient. It's this charge difference just across the lipid bilayer that really matters for this voltage sensing portion or voltage sensitive portion of the membrane spanning segments. So when this potential changes, it's going to trigger a conformational change in the protein that isn't really set up like this. But anyway, picture these long chain of amino acids, and when the membrane potential changes, so it's negative outside and positive inside, then it's going to put a torsion on these amino acids, and then that torsion is going to be displaced throughout. And it's like having like you shake a few links in a chain. That's going to pass through the whole chain, and then over some predetermined period of time, it ultimately is going to cause a conformational change in the inactivation gate, as well as the activation gate to open it, right? So the membrane potential here is one person in your circle of people elbowing the neighbor, going, open your mouth. They open their mouth. Now shout. The sodium's going to move. And then shut your mouth. Shut their mouth. And then they have to shrug their shoulders, kind of shake it all off, and rub their cheeks to get this inactivation gate closed again, and to reset the inactivation gate out there. So that's the whole series of events that we're going to think about based upon that metaphor that we used of the people in the circle screaming. So at this point where this is closed, we just have the biophysical properties determining the resting membrane potential, mostly potassium, so we're at minus 70 millivolts. Now what we have done is we have added positive charge with this electrode. So you see this electrode in here. We haven't done anything to it yet, so it's going to be cathodal, where it's going to add positive charges to the inside. So it's going to cause positive charges to come to the inside of the membrane. And when you get positive charges coming to the inside of the membrane here, the opposites are going to attract, so it's going to pull negative charges on the outside of the cell close to the membrane, and the sodiums will be pushed farther away. So are you seeing that in your mind? If you add positive charges experimentally to the inside of the membrane, then it's going to push away the positive charges on the outside of the membrane. Doesn't have to move them too far, and then pull negative charges in. And then what we experience as this membrane protein is that now it's negative out here and positive in here, and that triggers that conformational change. We got nudged by somebody's elbow, and so we now open our mouth, and the sodium can begin to move in. You see they're not showing the sodium beginning to move, but that's why we, as soon as we have a sodium permeability, a sodium conductance, a sodium current going into the cell, we get deflection of the membrane potential toward the equilibrium potential of sodium. And as more and more sodium moves in, you have a more robust or greater permeability. And remember, we said the permeability has no meaning without the conductance. So once you get the conductance, then you have a higher degree of permeability because things are moving and you have a current. Then you get a greater deflection in the membrane potential. In other words, more positives are showing up on the inside of the membrane, and more negatives are being pulled to the surface of the outside of the membrane. So you now have a complete reversal of the charge across the membrane. That's the membrane depolarization. It is opposite of what you started with. So it doesn't matter how long that would be there. Once you start this event of opening a sodium channel, you have a period of time before which this chain of events of conformational changes that opened the gate is going to get down here to the inactivation gate and click it into place and shut the person's mouth. Right? So they stop screaming, and they cannot scream again yet. They've got to shrug their shoulders and rub their cheeks. So there's an additional period of time before they are reset. And here again, they're showing one ion channel and this whole change from resting membrane potential to plus 30. Well, it's a population. So that's why I tried to introduce that earlier on. It's not just a single channel that we're dealing with to produce this measured event that we see here. So I don't like these reduction illustrations, but we have to use them to help make the point. But we have to use our brain to activate more circuits to say this would be, hundreds of thousands of these proteins within a region where we would have a big glass microelectrode coming in here and changing things. Then over a period of time, having nothing to do with our stimulating or recording electrode, we in this recording, this is actually when illustrations are missing something, it aggravates me. So this is the recording electrode way back here, but there would have to be a stimulating electrode that would put the positive there as well. They're not showing it. See, this what aggravates me about these incomplete physiological illustrations, because you're saying, well, how the hell did these get here in the first place? Well, there's a lot of different ways of doing it. We're talking about a depolarizing stimulus. Well, it's typically an electrical stimulus that will cause positives to come here and negatives, therefore, to come to the outside. Anyway, somehow we get the positive here, and then that pulls the negative to the outside, and it changes this. Once that happens, you have a certain time period over which this population of affected voltage-gated sodium channels will contribute to this profile of the measured membrane potential, and this is the intracellular electrode. Then there'd be a reference electrode outside, which isn't being shown because this membrane potential difference is inside to outside, right? So under resting conditions, it's negative inside versus outside, right? And now we're beginning to get it more positive inside to outside, and more positive inside versus outside, and the most positive inside versus outside as we're getting this action potential to be generated. And then as we begin to see the potassium contributing to the repolarization event, because the potassium channels are completely open and the potassium exits the cell, we are now beginning to get the final resetting. You see the activation gate is closing, and the inactivation gate is being reset to the open position. Then again, it is a population event. This was the first one to open, it'll be the first one to be reset. So if it opened at this time point here, it might be reset by this time point here. But the one that opens, the population that opens, the subpopulation that opens at this time are going to be reset at a time over here, right? So keep that in mind. And for me, actually, it makes more sense thinking of the population than looking at one channel going, well, what's going on here? How does that really explain the nice smooth curve? Well, it's the whole population of proteins having a biological behavior that produces a smooth, temporally related signal. So the electrical signals then are really dependent on the ion movement during the action potential. So if we begin to think of populations of sodium channels being activated, then being offset in the time that they start, we can picture that gradual event that is occurring. So we have some type of depolarization, whether it's a stimulating electrode that causes the outside to be negative or causes the inside to be positive, and then you get the corresponding change on the other side of the membrane. Right, so you can have a stimulus that would pull positive charges away from the membrane on the outside, then the negatives move in close, and then that's going to pull the positives on the inside of the membrane close to the inside of the membrane. Anyway, there's various ways that you can do it. Physiologically, we're talking about a graded potential getting to the trigger zone and causing a depolarization that would be at threshold or superthreshold levels at the trigger zone. So I like that idea better. Let's go physiological. And then the sodium channel activation gates will open rapidly offset a little bit spatiotemporally across the whole population at the trigger zone. So we get that smooth line rising phase, and then we begin to get the sodium entering the cell with the steepest slope of the rising phase representing the point where we have maximum number of opened sodium channels, and then they begin becoming inactivated with the inactivation gate over a period of time, such that by the peak potential of the action potential curve, all of them have been closed. So they talk about this positive feedback cycle where you get more depolarization that is going to open more channels, but not the ones that haven't been reset yet. They're going to be nearby ones, a nearby population. So I don't like that they came back in this direction to do it. But if we're thinking just of the sodium channels, the sodium entering the cell is going to have a positive feedback cycle and cause more depolarization for any sodium voltage-gated sodium channels that have not already been activated but are in the reset state and locally available to be opened. Okay, so again, thinking of the population and population dynamics of what is their present state is really important to understand even this illustration. And during the falling phase, to stop the cycle, the slower sodium channel inactivation gate closes, as we just saw. So that stops the sodium current, and then the slow potassium channels open, and by the time of the peak membrane potential change, at about plus 30, we have enough of the potassium channels open and a sufficient amount of potassium current as potassium leaving the cell that we get the repolarization, and of course it overshoots to the hyperpolarization to reach the voltage at which the potassium channels begin to close. So now we can put this in the context of this very nice illustration showing us the voltage in the voltage sodium and potassium gated channels. You can always recognize the sodium, because the voltage gated sodium channel, because it has the inactivation gate hanging off an intracellular portion of the membrane protein. And then the potassium is just voltage sensitive for opening and closing, so it's just shown as the blunt surface both outside and inside. Remember, if you looked from the surface, these would be complete circular channels. We have a longitudinal section down through them, so picture looking on the tube of your roll of toilet paper versus cutting straight down the center from top to bottom of your toilet paper, and then looking at its cut surface, you would see, the hole through the tube of the toilet paper roll, and then the roll of the rest of the toilet paper over here on the side. So just felt I needed to point that out. Again, these illustrations, I don't want them to be misleading. So we are seeing the channel itself passing through the center of the toilet paper roll, right? By looking at this longitudinal section through these ion channels. So it's blunt on both sides for the potassium, and then it's got this little inactivation gate for the voltage-gated sodium. So we have these represented at each point, and then we are talking about the excitability of the membrane. So this helps us add this portion to talk about the refractory periods, both the absolute refractory period and the relative refractory period, but there is the complete refractory period that starts at some point where the essentially the full population at this localized area, the circle of people that you're looking at, this localized spot on an axon or the trigger zone where you are measuring this event, observing this event. Farther down the axon or somewhere else on the axon hillock, it might not be the case, right? So keep that in mind, that this is a very localized event that we're looking at. And at that localized area, there is a population of these types of channels, and we are asking the question, on average, for that population, what is the likelihood or the probability that we're going to see a certain proportion of these channels at this state? And that is why we see a gradual event, other than the gradual change in the amount of the ion either on the inside or the outside of the membrane, or charge rather than ion, if you want to think of it that way. So again, we see the red curve with the membrane potential for the action potential and the ion permeability for the sodium and potassium. So we saw that those are the same, and on the same time scale, you can see that the entire action potential is spanning the point to full recovery about 4 milliseconds. But the rising phase to the falling phase, where it crosses again the resting membrane potential before the after hyperpolarization, is about 1.5 milliseconds. So it's relatively fast. So at any one point in time along here, we see that when the vast majority of the population of voltage sensitive or voltage gated ion channels are in the state where they can be activated, you have a high level of excitability of the membrane in that area. And then there's every range in between all of them being available to time points when none of them are available to be activated. And so when none of them are available to be activated, that's the absolute refractory period. Okay? And they're showing that as all of the sodium channels being inactivated at this point. So even though they all may not begin to open at exactly the same point in time, remember this is a population across a certain volume of the membrane, there is a point in time where they are all inactivated. While they are activated, they cannot be reactivated either. That's why you see this point where they are activated, open, and actively conducting the sodium ions included within the absolute refractory period. If it's already conducting, you can't activate it to conduct again. So you can't trigger another action potential while an action potential is in the rising phase. And the absolute refract at that particular portion of membrane. And then because the inactivation gate being closed is not the same as the channel having the capability of being activated, you're still in the absolute refractory period until you have the completion of the reset. And what I'd like you to do is to look at this, even though they're showing it as a temporal event, to complete the reset such that you have a voltage-gated sodium channel that is capable of being activated. I'd like you to look at this as a population. So at any one point in time across this whole spectrum, you ask yourself, what is the likelihood that I'm going to find a certain proportion of the population of voltage-gated sodium channels in this state, in this state, in this state, and in this state? Right? And so if we were looking at a single point in time where we had 1 out of 4, 25% of the population already reactivated, what strength of stimulus would we have to administer to trigger another action potential in this area? That's the relative refractory period. Hopefully that makes sense for you, right? Now they're using this to show a temporally, linearly related event of a single ion channel, voltage-gated ion channel, going through this reset series of events. I used it also to show a population within a segment of membrane, and within that population we see that they are all at slightly different points in the reset. And so if 50% were available, then you'd have to give less of a stimulus, stronger than what you did originally, but not as strong as you would have to when only 10% or 20% or 30% of the channels are available. That's what makes this the relative refractory period, that if you have a strong enough stimulus to open whatever proportion of voltage-gated sodium channels are capable of being opened, to get that number of people shouting again, you can get an action potential. It's not an efficient way of doing it. But also remember that you still have to reach threshold. So as I said, one way of modulating the relative refractory period is impacting the potassium channels. So if you can cause this, if you can increase the negativity of the slope of this potassium permeability and conductance curve, then you would get it back to baseline sooner. And so this portion right here can be modulated. This portion can be modulated too, but it's kind of tough to get into the absolute refractory period because you just don't have enough of the population of voltage-gated sodium channels proportionately available. Right? And there's not a large proportion that are going to be available back here either. But if you can impact this portion of the potassium permeability to get it back to baseline before the end of absolute refractory period, you can shorten the interval between action potentials. So if in this one segment of an axon hillock, or along an axon, you can, or on a portion of membrane, if you can impact this, you can actually increase the maximum rate of frequency, the time interval between action potentials is what differs for different frequencies, right? So if you have 1-2 milliseconds to play with to impact your maximum firing frequency, then you have an additional range of peak excitation to deal with. And you don't typically see that type of modulation going on in areas other than the higher levels of integration within the CNS. But I want you to understand that these are properties that can be impacted by molecular mechanisms. And what they do is they change, they modulate the excitability of neurons such that you can have an enhanced area on the upper end of signal intensity to codify in the frequency modulation and interpulse interval aspects of the action potentials going down the axons, right? So I know that makes it pretty complex for you, but this is the way you have to think about neural integration. You have to take the simple and push it to the extreme. But in general, for the general somatic efferents and the general somatic afferents, you're not going to have much modulation of the relative refractory period. So then the minimum time interval between two action potentials based upon refractory period is going to be 2 milliseconds. So from the or actually you would say 4 milliseconds from the time point of beginning the action potential. So how many sets of 4 milliseconds can you fit into a second? That's still a pretty high frequency, even if you're playing with that. But again, remember that typically you don't get a 100 hertz signal that goes on for 60 seconds straight. You're going to have a burst or a pulse of a set of action potentials that are at a frequency within that pulse of 100 hertz. And then you're going to have an interpulse interval to where the axon is silent. Then you're going to come with another wave or pulse of action potentials. So understanding this relative refractory period lets us appreciate the maximum firing rate for a neuron. Now, for cardiac myocytes, it's going to become very important to understand this in greater detail. And there is a lot of modulation of the potassium such that we extend the action potential so that its time interval equals the contractile event of the cardiac myocyte. You don't want to be firing another action potential to contract before the cardiac muscle cell had a chance to rest, right? Otherwise your heart's going to go and stay there. So it's at this point that I introduce the modulatory capabilities so you can appreciate that now as part of the ability of neurons to modulate signals, and then later why it's important for cardiac muscle tissue. What I'm going to do for this illustration is to go to the next panel, panel B, first, so we can see what's being recorded here to make sense of it. But what we are considering is the coding for the stimulus intensity. And this illustration helps us to make more sense of it. Okay, so what we're talking about is an anatomical site in a neuron called the axon hillock, and a functional or physiological site called the trigger zone. And that works great for a motor neuron, where the axon hillock is the beginning of the axon, close to the cell body, and the action potential is going to originate from integration at the cell body and go down the axon away from the soma. Well, for a pseudo-unipolar neuron, this is totally different. The trigger zone is in a completely different anatomical localization, as you see here. So this is the cell body of the pseudo-unipolar primary sensory neuron, and then it has this primary process off the soma that divides into a central process that is going into the CNS. They show it short here, but it's actually longer. It's just the illustration. At the end of that axon, you're going to have the classical, the central process will have the classical axon terminals with vesicles containing the neurotransmitter. And many of these are glutamate, a fast excitatory neurotransmitter. You want your sensory signal to be transmitted fast to have major excitatory effects on the target neurons. Okay, so where's the axon hillock? This is the initial segment, but that's not the axon hillock. So we don't use that term for this. Okay, so if we don't have an axon hillock, where do we have a trigger zone? Well, the trigger zone is that site on the peripheral process where we are going to have the initiation of the action potential, and in order to satisfy that criterion to be the initial site for generation of an action potential, we have to have the highest concentration of voltage-gated sodium and voltage-gated potassium ion channels, as well as, of course, the sodium-potassium ATPase pump that's going to be differentially located. You want more of that around to help get back to resting membrane potential, where you're going to have the larger proportion of these types of events. Anyway, I just don't always want to leave out the sodium-potassium ATPase pump and how it does contribute to resetting the resting membrane potential. So if we had some way of visualizing voltage-gated sodium and voltage-gated potassium channels, and it generated a fluorescent signal, such as an immunofluorescent type of a technique using antibodies to those, then the highest degree of fluorescence would be right here at the trigger zone. But we'd also see a very uniform high fluorescent signal all along the rest of the peripheral process, and definitely on the central process, as we would expect on any axon, and then on to the terminal. And if we had a fluorescent antibody for voltage-gated calcium channels, it would be right here at the axon terminal, because we need to have the action potential converted into an exocytotic vesicular neurotransmitter release event, and that depends upon calcium, so we've got to have the sodium conductance be converted to a calcium conductance, so we get a localized calcium here to get the exocytosis. Okay, so that explains what we have there. And what type of ion channels we would see at what particular density all along here. And then it would be kind of like a dendrite in general, different populations of membrane ion channels here, so we could have graded potentials to get integration at this most distal portion, most remote portion of the peripheral process. So we will have some type of receptor, whether the afferent neuron terminal itself, like a free nerve ending for pain, or, we'll talk about the different types of sensory receptors, but there's got to be a receptor mechanism such that the stimulus will trigger a graded potential by whatever means in the the terminal portion of the peripheral process of this sensory neuron, primary sensory neuron, which anatomically happens to be pseudounipolar, right? So if we measure there at the first part of the event, we will see, and this is going to be time as well as distance along the peripheral process that we're going to see these events occurring. So that's why I wanted to show this one first, and then go back to the previous one. So a stimulus at an intensity that would cause a deflection from resting membrane potential of minus 70 millivolts to approximately 0 millivolts will, by the time and at the distance it reaches the trigger zone, still have sufficient amplitude to be above threshold, but it is higher above threshold such that not only an amplitude, but sufficient amplitude to stay suprathreshold for a longer period of time. So the width, if you will, if you're going to measure the distance, it's also be the time interval over which you have the membrane potential greater than threshold, is greater than in another instance, you're going to fire more action potentials over that time period. Right? So this time period may be the same from this point to this point, from resting membrane potential at the spike zone to resting membrane potential again. That might be the same time, but of that total time, how much time is the membrane potential above threshold? Right? So less time is spent above threshold for this one. Threshold must be a little lower, I guess. Then you can generate a certain number of action potentials. So over this time period, how many of these events are contained within that time period? That generates the frequency, right? So here we see the frequency being initiated at the trigger zone. So 1, 2, 3, 4, 5, 6, 7. So if this was a 1 second time period, this would be a hertz of 7. If this was equally a 1 second time period, we would have 1, 2, 3, 4. So this would be 4 hertz. So this stimulus intensity would generate a 4 hertz pulse. Now we're not considering an interpulse interval in this one. We're just talking about generating a single pulse, a collection of action potentials that's going to have a certain frequency. This is 4. We're saying that this is a 1 second interval. And then in the axon, the central, the more proximal peripheral process, but also in the axon here. Once we get it into the peripheral process here and it bypasses the body and gets on the axon, whoops, I jiggled my thing and it jumped to the next slide. Or even if you're measuring right here in the axon terminal, you're not going to change this frequency. The amplitude of the action potential and the frequency and the interpulse interval, once 2 pulses, back to back are sent down, you're not going to change any of that. That makes it a digital signal. It's not an analog signal. It cannot be changed in amplitude, the height, and the extent to which you get the change in membrane potential, the amplitude, and any distance or temporal relationship within that signal cannot be changed. It's codified. It is a burst was it 7? 1, 2, 3, 4, 5. Did I miss one? 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7. Oh, they messed up on. You can't add an action potential. However many you generate here in one pulse are the same number that have to go on. So somebody didn't count there, or maybe I missed one. But anyway, the number that's generated here is the number you're going to see here, and the number you're going to see there, and the number you're going to see here, and the number you're going to see here. And that determines how many voltage-gated calcium channels are going to open, and exactly how many vesicles, presynaptic vesicles, are going to fuse to the presynaptic membrane and undergo exocytosis. And you assume the same number or same concentration of transmitter molecules within every vesicle, so a certain number of vesicles will fuse with a certain number of action potentials, and that's how much transmitter is going to be contributed to the fluid in the synapse, and then you convert digital to analog, because you can have any range of chemical concentration in the fluid. Signal transduction, again. So this is the way that it will work for a sensory signal. Now you can change the trigger zone to the axon hillock for a multipolar neuron, an alpha motor neuron, and be the same way. Graded potential out on a dendrite. If it reaches the axon hillock like this and generates that many action potentials in that pulse, then that pulse will go down that axon all the way from your primary motor neuron in the cerebral cortex to the lumbar spinal cord or sacral spinal cord to for that alpha motor neuron, or from that alpha motor neuron all the way to the muscle in the bottom of your foot. That number of pulses is going to go down the axon. Here, with a sensory neuron, we're just talking about it being generated at the trigger zone in the peripheral process, and it's going to go all the way into the CNS to the axon terminal. So here you see that the integrated stimulus intensity in the integrating portion of the neuron I want to say converges, but I don't want to use that term. Reaches the trigger zone as an integrated data set or collection of information, and makes a determination that is ultimately expressed as a pulse of a certain number of action potentials over a certain period of time, hertz. You get a frequency of firing for that neuron. But it's not frequency alone, it is interpulse interval as well, as we said. Okay, so then to summarize that a little bit, I guess, for the trigger zone, wherever you're located, the graded potential enters that zone, your circle of people that are going to shout, open their mouth and shout. the voltage-gated channels open. Of the population, there's a gradual change in each of the persons, and they all are going to begin opening their mouth when it happens, and it'll be fast. The voltage-gated sodium channels open. Shouting begins, slightly offset from person to person. The positive charge spreads along adjacent sections of axons by local current flow. So there's adjacent circles of people, and when one circle shouts, the next circle is going to shout. And then we begin to see the generation of the conductance of the action potential. The local current flow causes a new section of the membrane to depolarize. Then the refractory period, so if you get circles of people within a parking lot, it begins to spread through the parking lot, right? The refractory period prevents backward conduction. Loss of potassium repolarizes the membrane. So where this gets confusing is to show an image of a segment of an axon or of the trigger zone here, and to kind of confuse a local electrical stimulation to depolarize it versus a natural one coming through an integrated set of graded potentials. So if it was an integrated set of graded potentials and this was the trigger zone, we'd kind of imagine them coming in on the left here and not hitting it right in the center. It can't do that. So this actually is an illustration of the trigger zone where they applied a local stimulus such that it accumulated positive charges on the inside and as a result negative charges on the outside, or somehow they applied a current on the outside that was negative that pulled the positives away, and then it's like, well, if you guys are going to move away, then us negatives are going to move in. If the negatives move in, then on the inside of the membrane the positives go, well, you negatively charged proteins, get out of the way. We're going up to there because we like those negative charges out there, right? So however your electrical current, electrical stimulus is applied, it results in the same thing. Positive charges on the inside of the membrane and negative on the other. Well, that's the opposite of resting membrane potential, so you have a localized, depolarized event. So you end up with a depolarized section of an axon. Okay, so they call it trigger zone because if you electrically stimulate any portion of an axon, you have converted it, therefore you've forced it to be the trigger zone. Physiologically, in a multipolar, in a typical neuron, everything excluding the primary sensory unipolar neuron we just saw, the trigger zone is at the axon hillock, or that other exception that we just saw. But still, physiologically, the integrating center is going to be remote from that, and you're going to have the analog graded potentials integrating until the trigger zone is affected. But if you just come in down here, say here, and this is a stimulating electrode, then you force this to become the trigger zone. And what's going to happen to the action potential? Well, it can go this way, and it can go that way. It's going to go wherever it can, wherever the sodium channels are available to be activated. So to go back to our parking lot of little circles of people available to open their mouth and shout, if you go right into the center of the parking lot and begin to elbow people, nudge them, then if they begin to shout, then it spreads in every direction through your parking lot. And of course, that's not the way that you want the biology to work. The way you'd really want it to work is to be at the edge of the parking lot at one location saying, I'm the receptor, and I'm going to cause the trigger here, and then when these guys start, it's going to go just in that direction, because after this circle shouts, they have a time period where they need to reset, and then the potassium is going out, so they can't begin to shout again. So when this group circle activates, they can't send the signal backward. They're trying to, but they can't, because this circle group is just not ready to do it again. But on the other side, they're ready, and it continues on like that. So that's the concept that we want to build on now. And I don't like it when they have trigger zone here and they talk about artifactually forcing a segment of an axon to be a trigger zone. But anyway, wherever you start the depolarizing event, that now is your artificial or categorically your trigger zone, right? And that's this property, this active property, all or none property of the action potentials. Wherever you start it, I'm going to spread it. And if you physiologically are controlling it so it's unidirectional, great. That's the way that we're going to do it. But I'm just a voltage-gated sodium channel. I'm here ready to be elbowed and shout. That's it. That's all I do. And whoever's around me hears the shout, and they're going to respond the way that they can. If they can't respond, they're not going to do anything. If they can, then it's going to spread that way, and it's going to spread that way. So let's get to the physiological one, with a trigger zone at the axon hillock, the one that we typically think about. We saw the exception first for the sensory neuron. Now we're putting it back at the beginning of the axon for a multipolar neuron, say an alpha motor neuron, or an interneuron in the cortex, a large pyramidal neuron. Then all the integration of the input occurred at the dendrites and the soma, and you had the analog graded signals reaching or congregating finally at the trigger zone, and they either reached threshold and would hold that super threshold for some period of time or not. If they reach that threshold, then they are going to initiate the opening of a subset of the population of voltage-gated sodium channels, and it is going to be enough for that subset to trigger the nearby subset, but it is unidirectional, because where it first starts, it cannot come backward. So here we see the graded potential above threshold reaches the trigger zone, and we get a population of the voltage-gated sodium channels open. We're just going to think about sodium channels. The voltage-gated sodium channels, I think this goes in the sequence here, they impact their neighbors, and so the nearby voltage-gated channels open, and the sodium enters the axon in that localized region. But it cannot go backward because this membrane has not reset enough to be depolarized again. It is still in the absolute or relative refractory state. And so I don't even like that they put it there, but the influence is still there for it to go backward. And they're already showing that this membrane has reset to negative inside and positive outside. So why did they do that? It's aggravating me. Then the positive charge flows into the adjacent section of the axon by the local current flow. And because it can't go backward, it is unidirectional away. So when it initiates at the trigger zone, it cannot go backward. So the trigger zone doesn't reset within the time frame of this local event, so it will not go backward. Just scratch that from here. Why did they do that? Ugh. Okay. And then you have a resetting membrane. See, why are they showing the negatives in and the positive out? So it's not back at resting membrane potential, or even if it is, it's still within the refractory period, and they cannot be reactivated. But these arrows are showing that the influence is there. If enough people were ready to shout, open their mouth and shout again, you could get it there. But it does not occur. So the more proximal portion of the excitable membrane, either the trigger zone or the axon itself, is in refractory period, so it cannot experience another action potential. So obviously this continues on distally from active region to inactive region and leaves behind it a trail of refractory region that cannot be reactivated. So another way of looking at this is to think of a series of recording electrodes along an axon, and we're thinking of the normal physiological origin of the action potential proximally going distally along the axon. So electrode number 11 is the most distal and number 1 is the most proximal. And of course on the y-axis we have membrane potential in millivolts and time on the x-axis. So at any one point in time at this location, so at this location we are thinking of time 0 to time 4 milliseconds, right? Within 4 milliseconds we're going to see this full pattern at number 1. At number 2, within that 4 milliseconds, we are going to see, well actually it's more than 4 milliseconds. No, this is 4 out to here, right? So at about 2 milliseconds we're going to begin to see this portion, and then at 4 milliseconds we will not have seen the full action potential there, and then a portion of one at 3 and a portion at 4, and then as we extend farther out in period of time we're going to see partial features of the change in the membrane potential as this action potential is moving from proximal to distal locations. So this is kind of a difficult thing to picture because we are superimposing time on distance, and that's kind of a difficult conceptual thing for the human brain to do. But what we want to realize is that as soon as enough of the membrane has changed its polarity, enough people have started shouting, an appropriate proportion of the nearby voltage-gated sodium ion channels have changed here, they will begin opening, elbowing a group here to start opening their mouths and to shout. So at any one point in time you can see a portion of the action potential wave, and the earliest portions will be occurring first. Anyway, but what's more important is that we look at this here. When at the time point, remember this is time, at the time point, roughly about 4 milliseconds, what was it, like 2 milliseconds to, or 1.5 milliseconds to the 4 milliseconds, the refractory period. At 1.5 to 4 milliseconds at this location, when sodium channels are opening are opening and opened, and there's a lot of sodium current, and the membrane is depolarized at position number 2, position number 1 is in its refractory period. And so you cannot have an action potential at number 1 at the time point when you have sodium changing the polarity of the membrane at position 2. So at this physical location, temporally, the refractory period overlaps the depolarization phase at position 2. And like I said, it's kind of challenging, but that's the way you have to look at it. So in other words, the simple basic conclusion is that normally, physiologically, the action potential is unidirectional. and therefore we always learn that the axon is that portion of the neuron, that process of the neuron that carries the information away from the cell body. It's a unidirectional flow from the cell body away to tell the target cell, hey, here's what's going on. Here's my codified signal. So let's now think about the speed of the action potential. And again, we're talking about a pulse of action potentials. And then they begin just talking about a single action potential because that's conceptually easier. But remember, where you've got one, you're probably going to have a few more in that pulse, unless it's a 1 hertz signal, right? So if it's a 1 hertz signal, then it's a 1 action potential in a second, and that's that pulse. So let's think of a 1 hertz signal, a single action potential within a second, being conducted down an axon. Then we can think of the speed of a single action potential. But if it's a pulse of 7 or 8, like we saw before, they're all moving down the same way. So the same basic property is going to apply to each of those action potentials as they're moving down or being conducted by the axon. So the speed of the action potential in the neuron is influenced by the diameter of the axon. The larger axons are faster, and the resistance of the axon membrane to the ion leakage out of the cell. And the myelinated axons are faster. The myelin actually acts as a capacitor. And I'm just going to convert my pointer to a felt tip to describe what a capacitor is. So a capacitor is 2 plates. We consider these as some type of a source of a charge here, right? To try to draw this circuit. And these are 2 plates of the capacitor, or this would be the heads of the phospholipid bilayer, and then these would be the little legs. The tails, they're called, right? So this would be the lipid bilayer. So you've got the 2 plates that are separated by the, it's about 5 nanometers for each of these. So I think it's 10 nanometers total. Should have made that 10 nanometers total, but it's the length of a phospholipid is 5, so 5 and 5 is 10, right? So the 10 nanometers is the distance. So it's really close together. That makes it a high-strength capacitor. So a lot of membrane potential whenever you have collection of charge on each plate. So normally, the resting membrane as a capacitor has positive on the outside of the membrane. I should always label these, and negative on the inside. So this makes the membrane a capacitor. The resistance of the membrane is determined by the ion channels, right? I'm trying to show the channel here. And then illustratively, I think our resistor is like zigzag, isn't it? Something like that. So you've got the resistance. So this is the resistance, and this is where you can have the current flow. And the capacitance is determined by the distance of separation. Okay. Now let's put the axon here and add the myelin sheath to it. Right. Let's say this is in the periphery, and there's the nucleus. Then on the other side of the axon, remember we've got a longitudinal section to this. Okay. So now what we have done is we have separated by a much greater distance the charges across our 2 plates on our capacitor. And as you increase that distance, you reduce the capacitance. I'll let you write capacitance there, capacitance. So yeah, I'm not going to hold you responsible for all these electrical properties, but it's important to realize that you separate the charges, you reduce the capacitance, you in effect also reduce the resistance to linear current flow. Okay, so what types of current flow are we talking about here along this membrane? Oh, I converted this membrane into an axon over here. Sorry, cancel this part of this picture out. Sorry, it would have been negative charges over here, right? Not there. Oh, I hate it when I do that. Anyway, these negative charges would still be down here. Don't put them there. Sorry about that. But the positive charges would obviously be moved farther away. So if this is the membrane, then this is inside the cell here. So if these are all sodium molecules, sodium ions inside, as we will see, they can flow along the inside of the membrane. And there's less resistance if you have a greater capacity. And then, so we'll see this again when we look at the conductance in a myelinated axon. But it's this point when they're talking about the diameter of the axon and the resistance. So the greater the diameter, the lower resistance. So then you're going to have a lower resistance to the linear flow of current along the larger diameter axons. But you can't, it's just impractical to have gigantic axons. So you have to have smaller ones. So you have to increase the capacitance with the myelin to have the fast conductance rate. So the myelinated axons are faster because the myelinated segments have a reduced capacitance and therefore a reduced resistance to linear current flow. So the current flows very quickly, whoosh, without even having to depend upon the voltage-gated sodium ion channels. And we'll see how that works when we get there. So here we are. So you see they show this sign here, the depolarization going linearly down through the axon. You're going, well, I thought the only way that the current flowed along an axon was where we would perceive the inward flux, the current of sodium. That's the current flow along the axon. Well, it isn't. Any place you have a recording electrode, remember I said that before, you have a recording electrode, and if you have a sodium with a positive charge that moves like that, right? Wouldn't move that far, but anyway, if there's a bunch of sodiums all lined up and they move like that thing just shot, and you're here, just like you saw it, you perceive a current flow, right? So if you perceive through measuring it here that a positive charge moved, that's a current, right? So whether it's a current going in there or a current going along the length, it's a current. Anytime you move a charged ion, that's a current. So think of it again as this population of sodium ions. They are outside here at the node of Ranvier. And when the membrane here is depolarized sufficiently, they will open. So the highest concentration is actually right here on this proximal side of the node of Ranvier. So it's most sensitive, most excitable. So any depolarization wave that reaches it will open enough of them to get everybody shouting here on the node of Ranvier, right? And so what's happening is just enough sodium comes in here to displace sodiums, right? I'm not saying that the sodium that is here ends up over here. What I'm saying is that there's enough sodiums that become pulled close to the membrane, and then therefore negative charges, sodium gets displaced out here, and negative charges are closer, right? So this sodium right here doesn't end up there. They just move short distances, but all they have to do is move. If they move and then get more of them close to the membrane here, then you've got, and I should make these all pluses, right, if they're sodiums. I was thinking of them moving linearly. So this sodium moves that one, moves that one, moves that one, moves that one, moves that one enough so that it pushes, and you've got sodiums here, and you got sodiums there, and then because the plus is there, the negatives come in here, right? So you get the negative charges here. You've got a locally depolarized membrane. Just enough to open this, and then you get the same type of reversal. So this is what they're, and they show this arrow going that way, pluses, and then this arrow going this way. You're saying, well, what does that mean? What's moving? Something's moving from here all the way to there? No, it's like being in a crowd. We now have the negative charges, the positive charges will be pushed back here to complete our circuit. So we got the whole flow, right? These, again, I'm not making all the pluses, but just picture the little shift of all the people that way, and then a shift of all the people that way. These guys get shifted away. Why? Because negatives come in, right? So if positive comes in here, pushes that guy away, pushes that guy, pushes that guy, pushes that guy, right? And then because he gets pushed away, a negative comes in because there was a positive. Now there's a positive there, and the negative wants to get close to the positive. And so if we think of it at that minute scale, it begins to make sense. This never made sense to me until somebody explained it on this level. So hopefully my explaining it helps you understand it that way as well. And so this becomes very rapid. All you're doing is like bumping enough of these along to create a current to get some sodium squeezed into here, and the negatives moved out of the way, and negatives pulled in, and then positives pushed away to basically displace back to here and complete that circuit. And this is very rapid. It won't go for long distances. The length of an internodal segment is about, as far as I understand, the most effective distance that you can get this type of a linear conductance of current, basically through the cytoplasm, right? And through the completion of the circuit through the local interstitial space. And remember, if you're in the CNS, you've got astrocytes right here. So this space is not a large space. It's not this big white empty space, or a satellite cell if you're in the peripheral nervous system. And the satellite cell or the astrocyte is going to, remember when we saw that picture, there's no surface exposed. So it's a very small space here. So it's not this big gigantic volume. It's the very restricted space, and this is how biochemically the other glial cells in the CNS and the PNS create this environment where these types of things can happen. You can get this little bumping of sodiums to complete this external part of the electric circuit to get a current flow through the cytoplasm here, and that's very rapid. And it can just kind of bounce back and forth and reset very quickly. Can't get it over long distances, but just enough to jump from node to node. So you get that saltatory conduction that you are certainly aware of. I'm resetting my pointer now. So in cases where the myelination deteriorates to whatever degree, they're showing a complete degeneration of the myelin sheath here, but it can be a partial one too. Any disruption of this specialized arrangement is not accommodated for by the insertion of sodium channels in the internodal segment. So basically what you lose is you lose that low resistance because of the low capacitance. You end up with high capacitance again, and you have a high resistance to the flow, so it becomes very, very slow conduction in the internodal segment whenever you have, or you get as myelin is deteriorated or affected negatively by whatever process, can be a toxin, the capacitance will increase, and the resistance will increase, and therefore the rate of conduction across the internodal segment will decrease, and therefore overall conductance down an axon will decrease. So there are ways of testing. You can electrically stimulate one portion. It can be proximal or distal part of the axon. Typically it's a peripheral nerve that you're doing this with. And some distance away you measure for the response, the action potentials to get there, and you know your distance and you know your time, and then you can calculate conduction rate. And the known normal conduction rate is well established, and anything slower would indicate something that's compromising the current flow along axons. Now we finally can talk about the chemical factors that can affect the electrical signals. So the effect of extracellular potassium is the one example that we're going to consider. The textbook mentions a few others, but we've been talking long enough, and I appreciate that you're sticking with this. So let's think about how potassium concentration in the extracellular fluid compartment, which is interstitial fluid and plasma, as the major components. The cerebrospinal fluid is another. The extraocular fluid, the ocular fluid, intraocular fluid, the fluid in the eye, and the fluid in the ear, anywhere, joint fluids. Those are all minor components of the extracellular fluid compartment. The interstitial fluid collectively of the whole organism, the plasma, and the cerebrospinal fluid are the major components. But typically when you hear extracellular fluid, you think of plasma and interstitial fluid. And they will be virtually the same at any one point in time and at any one location. So when the blood potassium is in the normal range, normokalemia, and that is, I'm going back to my textbook, 3.5 to 5 millimoles per liter is the normal potassium plasma or blood potassium range, so normokalemia. A subthreshold graded potential at any one neuron. Remember, if blood potassium is normal, then any neuron is going to have that same amount, that normal amount of potassium. You would expect to find that normal concentration of potassium on the outside of that neuronal membrane. So wherever you would do this, you would expect to see a subthreshold graded potential that would not fire an action potential. So if you were stimulating the cerebral cortex, a particular intensity would not trigger an action potential of that upper motor neuron's axon down in the spinal cord. So there's ways of doing these kind of recordings, especially in animals, and some clinical ways too. But theoretically, at least. Then in normokalemia, a superthreshold is going to trigger an action potential. So, okay, we got it. In hyperkalemia, an increased blood concentration, potassium concentration, brings the membrane closer to threshold, and now a stimulus that would normally be subthreshold can trigger an action potential. So here's resting membrane potential. You already see that it is not at minus 70. So what happens with hyperkalemia? The Goldman equation is applying, and it's not the permeability of potassium that has changed, it is the relative concentration inside to outside. So resting membrane potential is going to be higher, closer to threshold. So any stimulus that would normally be subthreshold now might be threshold and trigger an action potential. On the other hand, in the opposite direction, hypokalemia, again the Goldman equation, decreased blood potassium concentration hyperpolarizes the membrane and makes the neuron less likely to fire an action potential in response to a stimulus that would normally be above threshold. So what this means is that a critical value or panic value for potassium below 3.5 or above 5 is of great concern, because you neither want a hyper-responsive or hypo-responsive nervous system, and of course the impact on the cardiovascular system is even more dire. So I believe this is the final slide. So again, I appreciate your time and attention. And what we see here is this trigger to play the interactive physiology animation, Nervous System 1, the action potential. If you have access to the interactive physiology supplement to this textbook, I strongly encourage you to watch that too. I know it's a lot of time, and I appreciate that you took your time to watch this. But that adds some nice, very brief practical information. You have worksheets and quizzes there, and you would find those very useful.